Good evening and welcome to this evening's Cambridge Conversation. My name is Ed Bulmore. I'm a professor of psychiatry and deputy head of the clinical school in Cambridge. And I can't see you, but I understand that about 600 or so people are joining us this evening, and it's a great pleasure in particular to welcome back alumni of the university. We're going to be talking about dementia, the Dementia Journey. And of course, you know, it is an enormous challenge in all of our lives. But there are also many reasons to think that we could be finding new ways of dealing with the challenge of dementia. There are a lot of moving parts, there are a lot of possible lines of attack. Tonight, we're going to focus on three particular angles that we're very interested in exploring here in Cambridge. The first is really the question of diagnosis. You know, what does it mean to say somebody's got dementia? What does it mean to say somebody's got Alzheimer's disease? Second big question, how, when we think about something as complex as dementia, the changes in the brain, changes in the mind, what that means for the social environment, for policy, what's the importance of multidisciplinarity, bringing things together to create a more joined up approach. And the final topic is going to be AI and big data. As computers get smarter, can they help us do better in dealing with dementia in any way? So those are our topics, and we've got three absolutely terrific speakers to talk to you and with you about those ideas. First, we have Professor James Rowe, who is Director of the Cambridge Centre for Frontotemporal Dementia and Related Disorders. Then we have Dr. Maura Malpetti, who is a Race Against Dementia Alzheimer's Research UK Research Fellow. And finally, Professor Zoe Kurtzy, who's Professor of Computational Cognitive Neuroscience in the Department of Psychology. So let me start with you, James, if I may. And just to sort of get us going, can you tell us one project you're working on at the moment that you're really sort of excited about in terms of helping us think differently about the diagnosis of dementia?
Yes, so thank you. Good evening. What we really aspire to is to have the same level of laboratory precision when studying people with dementia or risk of dementia as you might more easily get in a laboratory setting, whether that's with stem cells or transgenic mice models. But we're not satisfied with those laboratory models. We want to understand, diagnose, and know whether we're hitting the target in people who have the dementia or who are at risk of dementia. What's become clear is that the illnesses of dementia begin decades before symptoms first appear, and this opens up an amazing window of opportunity to be able to treat and to prevent and avert the course of the illness. But to do that successfully, we have to be able to measure these processes, chemical and cellular processes, but to do so sort of in this closed box of the skull in people who are alive and well. That puts a lot of emphasis on some types of scanning technology with different techniques, looking at behavior, what's in the blood and spinal fluid, and to bring all this information together to pick apart that cascade of cause and events from that root cause of the illness through a cascade of events that leads to symptoms and the devastating impact that many of the people online would have seen in their own families. And by picking apart that cascade with precision, we know where to intervene, okay, what to target. We can also use these tools to look downstream and say, what's the consequence? What's the benefit of a particular treatment? Is this candidate treatment something that looks likely to work, we should invest more in? Or should we say, you know, it's a good attempt, but it's time to draw it to close and focus our resources onto better prospects? So we can look downstream at efficacy. We can also look upstream to try and stratify or target those individuals who are most likely to benefit from a particular treatment. Yeah. So this precision of the individual, precision of the treatment, and having sensitivity to the outcomes of novel treatments is key. And that's been a really exciting development here in Cambridge over the last 5 years in particular.
Mm-hmm. Mm-hmm. So, I mean, it is exciting that you can predict the, the process of dementia. People might say, well, what are you going to do about it? If with the scanning and the more detailed understanding of, how these processes evolve over time, you can pick people up early, that's great. But aren't you going to need to be able to offer them something in terms of treatment or prevention? And I wonder where your thoughts are on that.
Yes, well, I'd say we're at a tipping point in the science around dementia. It's a very exciting time to be in research for several reasons. One is that there's no shortage of potential ways to treat and avert the dementias. I think they are utterly treatable in principle. They're no different from cancer or HIV or diabetes. We should aspire and expect to treat these dementias, but we need to do so in a smarter way, getting the right drugs to the right people and the right stage of disease. Ideally, that stage of the disease process before people are even aware of it, before symptoms. To think about prevention. We're very familiar in medicine to treat cholesterol and blood pressure and the like, to prevent heart disease and strokes. That should be the aspiration in the dementia world. We're not there yet, but where we are is for some of the dementias, particularly some of the rarer genetic dementias, we have already in trials some extraordinary new technologies to try and deactivate or neutralize those faulty genes. And I don't know which of those will be proved to be effective, but there's a real sense, a tangible sense of new treatments that will radically transform the individuals concerned. Where those work for treating patients, we can bring it forward to treating people at risk of those genetic illnesses.
So that's great. I'm going to ask Mara and Zoe to pitch in with any questions they might have, but just one quick one for me. You mentioned several times, James, when you've been talking, I've noticed you put an S at the end of dementia. You talk about dementias. How many different types of dementia do you think there are out there?
Yeah, so, well, but to come back to dementia, it is an umbrella term. It covers a whole family of illnesses. Alzheimer's is the most common of the dementias. It's around perhaps half to 2/3 of all dementias, but there are other forms like dementia with Lewy bodies, frontotemporal dementia, some rarer dementias like Creutzfeldt-Jakob that you may have heard in the newspapers that are rare. Now, they differ in their details, in their causes and their symptoms, but they have much in common, that they change our mental faculties of memory or attention, how we see the world, how we speak and have social understanding. And the key thing about a dementia is those loss of those mental faculties is to the degree which is disabling. It gets in the way of a normal and everyday life. But all those dementias begin in a more mild form. So there are milder cognitive impairments where people may have symptoms that are troubling, but normal life essentially is able to carry on. And that is, I think, where we are in medicine, in memory clinics, is picking up early symptoms. What we aspire to do, and I think again is in science, is to pick up these illness processes even one stage earlier, before symptoms are there, trying to get a personalized sense of risk, whether that's genetic or environmental or lifestyle risks, because of what one can do as individuals to reduce one's risk, what policymakers can do to reduce our collective risk, and to then think about targeting particular treatments if you're at higher risk. But all that journey rests on an accurate and early diagnosis.
Maura or Zoe, do feel free to chip in if you've got any thoughts.
Well, I might particularly come.
I'll lob in one more if I may, James. When you think about the range of different targets, therapeutic targets, drug targets that people are investigating for dementias, there's a range of things on there: amyloid antibodies, anti-inflammatory drugs, various other approaches. If you had to put, some of your own money, as it were, into a drug development project, what target would you pick?
I would do 3 things for 3 different dementias. So one is, which is immediately tractable, is to treat the symptoms that affect your survival. And some of us may have seen the work in the national press recently around restoring some of the noradrenaline, this brain's adrenaline capacity, has an impact on apathy, and we think beyond that survival. That's an immediate possibility.
Okay, so noradrenaline.
a sort of forgotten member of the dementia sort of process is to take a step before that. All of the dementias essentially have an aspect of inflammation. Now, the inflammation may not be the root cause of the dementia, but it's a feature, a driving feature once the dementia begins. And to slow down that inflammation would have an impact, not just on one dementia, but potentially many,
Right.
many of these dementias. So as a single target to slow all of the dementias, I think reducing inflammation, and we can carry that knowledge from one disease to another. The third step in Alzheimer's, we want to pick on really that most common, the most disabling of the dementias. I would look at the way that the buildup of this junk protein tau and its spread through the brain, how that disrupts brain cells, and the way that we can deactivate that process. So that would be a much earlier sort of preventative disruptive intervention to slow or halt the dementia processes. So it may be a combination of all three is going to be needed.
Right. Okay. So your top investment tips are noradrenaline, inflammation
and tau.
Great. Okay. Thanks very much, James. And that is quite a nice segue, I think, to Mara. Do you want to begin by just reflecting on some of the work you're doing at the moment that you're most excited about in relation to dementia?
Yeah, thank you very much, and good evening, everyone. I think James really opened my floor in a nice way, because one of the main projects that I'm working on is studying inflammation in different types of dementias. And in particular, I'm looking at different tools that can measure this inflammation in the brain and in the blood of people with dementia. And it's very interesting because James has already mentioned this, but this is a common aspect, something that is similar across all different dementias, and is something that could be really modifiable and maybe could stop the cascade that leads to clinical decline. And in our work, we showed that it's not just a passive bystander, but is an active accelerator of clinical decline in these people. So basically with my research, I'm trying to find these fingerprints of inflammation in dementia and to understand which type of inflammation, which sub-targets we need to focus on, who are the people that can benefit most from different drugs, and also which is the time window that we needed to intervene. So if preventative or a bit later in the disease.
And I mean, I think it's fascinating just to step back and see it from a slightly, from the point of view of somebody who's perhaps not so expert in the area as you. How would you explain what inflammation is actually doing in the brain of somebody that might make their Alzheimer's worse?
Yes, so basically when we think about inflammation, we can think about the immune system, and when in our body, for example, when we cut our hands, there is a reaction and there is a swollen hand, and you can see this reaction that tries to cope with the damage. And something similar happens also in our brain. And unfortunately, we cannot see that because, of course, we don't have, we cannot see that with our naked eye. But this reaction of our brain immune system can be seen with these special brain scans that James mentioned that we are using here in Cambridge. And these cascaded events like signaling, chemical signaling that I'm not entering all the specific details, but can lead and damage the cells in our brain, and which then cause also symptoms and different clinical manifestations.
So, I mean, you've touched on one really important problem, I think, challenge for the field is how do we actually measure inflammation in the brain? Because in patients, I mean, it's one thing doing it in experimental models, but in real life, how do you, and we do have quite limited options. And brain imaging is, looks very promising as a way of giving us that measurement that we need, not just to where inflammation is in the body, but where it is in the brain. And I wondered if you could just say a bit more about some of the tools that you're using or you're aware that others are using that can help us do that, can actually help us see the inflamed brain.
Yeah, definitely brain imaging is one. And as you mentioned, also like in Cambridge, we are trying to develop novel blood tests for inflammation that can be easily implemented in clinics, or like much easier also to monitor over time, as have been already also shown in other disorders like depression, for example. And I think like I would like to touch one of the topics that you mentioned at the beginning with this, because of course, dementia is really like changing very rapidly. And when we think about old-fashioned or classic dementia research, we just think about clinical medicine or psychology. While here in Cambridge and in the world, there is much more effort in an interdisciplinary approach in each topic that we are studying, for example, inflammation. So we are bringing together brain imagers with immunologists, with clinicians, with research nurses, engineers, physicists. And I think it's extremely exciting period for dementia research for this. So we can really learn one another to get to those best tools that maybe like if we just focus on one part, like brain imaging, we cannot reach, deliver that result.
Right. So this is international cooperation.
Absolutely.
At a large scale and multidisciplinary.
Absolutely. Yeah, like it's not just local collaborations, but it's like a national, international effort. Like for example, in Cambridge, we are leading, we are part of the leading groups of Dementia Platform UK, which is a national initiative and a collaborative effort to find a treatment for dementia, sharing data, not just with academics, but also involving industry partners.
Yeah, no, I completely can see that you need that kind of scale to really have an impact. And I just wondered where you thought, you know, where are the points where we could look to make early impact through the kind of efforts you're leading, you know, in terms of policy or other changes we could make?
Yes, that's very good questions. I think there are different aspects. Definitely we need to change a bit our mentality, and as a researchers, but also as audience and also as population. I think from the researchers' point of view, like for example, I'm involved in Race Against Dementia, which is trying to bring a completely different perspective, trying to think out of the box of the classic research, bringing Formula One philosophy and acceleration to give, like to deliver much faster all the results that we are aiming to. And in terms of like mentality in a broader population, I think we should really try to raise more awareness about dementia and the fact that like try to give more access to people with dementia and their families to contribute to our research and also to be involved in research. Because for example, in Cambridge, one of the perspectives that we really care about is like the families and the people with dementia that are volunteering for our research. And they are not just the key contributors of our studies, but also they are helping us to shape our research, sharing their priorities and their ideas. So yeah, I think we should just involve more people and think a bit, change a bit the mentality.
Yeah, no, that's fantastic. Okay, thanks, Maura. So I'll turn now to Zoe and ask you the same question, Zoe. What's a bit of the research project you're doing at the moment that you're most excited about?
Okay, thank you, Ed, and hello, everybody. Yeah, so we work a lot at the interface of brain sciences and computational science. And as Maura was saying, it's very much a multidisciplinary effort, looking at cutting edge developments, not only in neuroscience and clinical sciences, but also in maths, computer science, engineering. So what we have managed in a short space and time actually is to change the diagnostic pathway. And how do we do that? We borrow models, algorithms that are developed by our colleagues in maths and in engineering. We train these models with large data. And that's thanks to all of the patients that open their data for research. So a huge thank you and a huge call to everybody to openly share a huge resource, which is their data, their medical data for research. So the models, the algorithms are trained with many different data types. We heard from James and Maura about brain scans. We heard about cognitive and clinical tests. We heard about inflammatory markers. We heard about proteins like tau. We heard about amyloid. All of these data are accumulated. The algorithms start looking at them, and while each one of them by naked eye might not make much sense and might not help to increase the potential for precise diagnosis, the algorithms can put them all together and can learn their relationships, the really intricate and multiplexing relationships that then can identify patterns, and that allows us to start moving, shifting the diagnosis and the prediction of dementia much early. Right now, we're looking at patients that have mild cognitive impairment, which in some cases and in most cases will develop to dementia, but not in all cases. We know that 20 to 30% of these patients will not develop dementia, but a good number will develop dementia. And what we are looking is to predict from a single brain scan, a 5-minute brain scan, and some clinical scales, whether these patients will actually develop dementia or whether they will remain stable. And we have algorithms that can do this really successfully. It's really exciting because we are now implementing these algorithms in clinical research at Addenbrooke's. We initially worked with a lot of research data, but now we can see these algorithms actually making accurate predictions for patients. And we can also apply these algorithms in data from clinical trials. We can more precisely stratify the sample of patients that participate in the clinical trials, and hopefully making a difference to drug development and target development.
It's such important work, isn't it? Because you've got so many data points on thousands of patients. How big is the database you're working on at the moment, just to give us a sense of scale?
So we are pulling data across from many different cohorts and many different countries. And we train the models depending on their complexity in sort of hundreds of patients. We start with something like 400, 500 to thousands, up to 10,000. And of course, the more data we have, the more advanced our models can be, more sophisticated, and the higher the precision with which we will achieve this prediction. And our aim is really to go as early as possible before symptoms occur. We heard many times that dementia starts in the brain 10 to 15 years before symptoms occur. If we can predict that early, we can also intervene early. And maybe we will be able to identify the targets, the drug targets that can act that early, reducing actually symptoms and reducing the damage on the brain that then becomes reversible after a long period of time.
Yeah, and I mean, as you said, you have to use artificial intelligence to find these multiplex patterns, because no one single risk factor is that predictive in its own right. But I just wonder, for again, for the benefit of people that might not be sort of specialists in the area, what class of predictive factors do you see most emerging as most important, if you had to sort of pick a few key ones?
Yeah, so of course, there are some predictive factors that later on could be actually quite precisely diagnosed. For example, we heard from James about tau, but these, when we identify these, is quite late. So our challenge is how to identify targets very early on. And so when we look, for example, brain imaging can also be highly predictive, but later on, when we look at images very early on by themselves, by looking at them at the naked eye, they're not predictive, and they are not actually specific to different types of dementia, which is really important to identify with high precision so that we can treat accordingly. So this is really the challenge. We have some predictive markers. We have, for example, tau, which is highly predictive, but they appear much later when the disease has settled. Well, our challenge really is to identify markers much earlier on, before actually each one of these, as you said, is predictive, and before actually we can see the disease, of course, already damage on the brain by naked eye. And actually, we have a large program with Alzheimer's Research UK, where we are looking into training algorithms, training models with data like tau data that are highly predictive, but also with data coming from digital technologies like wearable data. And the hope is that when the algorithms learn these patterns, they learn, for example, to associate warning signs with highly predictive markers like tau, like amyloid, then potentially, then we don't need this highly predictive data, which is more invasive. We don't need to have patients undergoing lumbar punctures or PET scans, but we can have quite highly predictive warning signs just from digital data. And of course, it depends then, you know, on the pathway, on the clinical pathway and the trajectory of individuals. But if early on we identify some signs, we can we can proceed with interventions in lifestyle factors, then we can reduce the disease settling.
Yeah, I was very interested by what you're saying. So that you mean wearables and digital activity, sort of patterns of online use, that sort of thing. Those kind of data you think might be predictive of early cognitive changes before?
But by themselves, probably not, right? Because one can get the flu, right, and feel unwell, and your Fitbit will show that your heart rate is not that good today. That doesn't mean one has dementia. However, you know, when the models learn to associate this data with other more predictive pathology data, then these patterns are what the models can extrapolate. And when we're missing the pathology markers, that the models can say with some certainty that data from the digital data from wearable technologies will be able to predict. Of course, then we need follow-up, but we don't always need follow-up. So maybe, you know, there's no cause for concern, or maybe there is an early risk. And I think James earlier on spoke about genetics. Again, some dementias are very much genetically linked, and these are the cases where we will have targets much faster, but there are other dementias where genetics are not as straightforward, and we also know that there is that kind of always interplay between genetic and epigenetic factors, environmental factors, that plays a big role on whether disease will be expressed or not.
I would just come in just to clarify this issue about the multiplex, the sort of complexity of data. I think we're past expecting that for most people a single gene or a single lifestyle or even a single blood test will be sufficient. We need the tools, the technology, and it includes AI to bring together lots of information. You know, human beings are complex. We have lots of genes. Individually, they're quite weak in their effect. There are very few genes that give a strong, you know, individual person risk. But add together lots of the weak effects for many genes, it starts to build up. So this is a role for AI to build what we might call polygenic risk, the cumulative risk from my unique combination of genes. It's different from other members of our family and other you know, everyone else in the human race. And next is human lifestyle is complex. Education, smoking, hearing loss, blood pressure, diet. No one of these factors individually has a big impact, but collectively it adds up. In fact, it's even some of the a dozen everyday lifestyle factors. I mean, hearing loss is one, education, pollution, you know, diet, exercise, and so on. A dozen everyday factors can explain around 40%, things that would be treatable with current technologies. But what we need is to bring that information together. And then the more advanced technologies, whether it's your phone or the wearable technologies or how we interact with computers. Again, there's not one keyboard button that gives us a diagnosis, but the whole pattern of how we interact with the world. So this complex, what we call multivariate, sort of gathering of multiple sources of information is going to be key. And that's really where AI comes into its own.
Okay, thanks, James. I'm going to come back to Zoe just for one closing question on your area, just because I had it in my mind. This definitely sounds like the future. How close is it to reality now? I mean, could you imagine running an AI algorithm in the NHS?
Yes, absolutely. It's definitely the future, but it's becoming near, and it's not science fiction anymore. About 10, 15 years ago, it sounded like science fiction, but now it's actually reality, and it's really up to us to open to these approaches and to make them happen. And really the key point is exactly what you said, Ed. It's really this translation. How do we move now from what we have in research to adoption within the NHS? And here we have some kind of challenges. For example, personal data security and safety is paramount. We need to respect this, and we need to be very mindful of it. So we have made our systems within our healthcare systems so secure that it's actually quite hard to put any new systems like software, for example, like these algorithms. But that's why we're working very closely, for example, at Addenbrooke's with a trust to be able to create solutions where data remains very safe. Nobody sees this data. Myself or my research team do not see this data. The data also is fully anonymized, no personal information, but the algorithms can reach this data. The algorithms can make the predictions, and then these predictions are interfaced in the form of scores and advisory scores to clinicians. Right. Currently, we are working in the space of secondary care, but we're looking into primary care also, and advisory scores can be given to the clinicians that suggest the potential prediction and risk, and then the clinician, together with other data, will be able to determine what is the clinical pathway going forward. Right. So we're not far, but still a few challenges to overcome.
Yeah, thanks. Very exciting to hear how close that is to real life, and also so important to emphasize the data confidentiality issues, because without trust, nothing happens. So thanks for that. Now, we have some questions, I'm glad to say, from the audience. And the first one is to Mara. And it is, I wonder if you could comment on the links between inflammation and dementia you mentioned. There's also, of course, some links between inflammation and depression. What are the links between depression and dementia? How do you see that whole triangle?
Oh, this is a very good question. Well, like for some types of dementia, and I think James as clinician can also feed a bit more, and this also add himself, but like for some types of dementia, some symptoms that resemble depression can be early signs of what then follows with other symptoms and different diagnoses. And I think like inflammation is a common pattern in both these conditions, but potentially they interact with different aspects. Like, for example, inflammation in dementia interacts with junk and tox proteins building up. So I think like we can share the tools to measure inflammation, but then when we want to intervene, we need to consider different aspects that create differences between these two conditions, and not just treat the inflammation in an isolated way.
Mm-hmm. Okay, thanks very much. We've got two other questions, or bunches of questions actually. One of them I call nature, one of them I'm going to call nurture. And I'm going to ask James on the nature question and Zoe on the nurture question. So the nature question, James, is about heritability. You mentioned genetics a lot. To what extent is dementia heritable? Are some more heritable than others, for example?
Yes, so this comes back to dementias plural, it's the family of illnesses. If we think about the most common dementia, Alzheimer's disease, it's actually very rare that a single gene mutation is a sufficient cause, a strong cause of an individual Alzheimer case, perhaps 1 or 2%. Now, there are genes, one you may have heard of called ApoE, that isn't a cause, but it does increase your risk 2 or 3 times. So it's not enough to get the illness, and plenty of people who have that gene variant don't get dementia, but it does increase the risk. And then we move into a stage where there are many dozens of genes that each give a tiny fraction of an increased risk. Not enough to say in any one person that that's the cause, but cumulatively, if I as one individual, or Ed, you as another individual, have lots of these weak genes, it starts to add up. So for a single gene that you would see passing through the family tree, that's very unusual. And yet it's also true that there is a strong, I suppose, genetic contribution to the whole dementia story. Mm-hmm. For some rare dementias like Huntington's disease, it's always genetic. For frontotemporal dementia, it's perhaps another rare dementia. Maybe a third of people, 20 to 30%, would have a faulty gene as the cause. But for the much more common dementias like vascular dementia and Alzheimer's, it's 1 or 2%.
Right.
If I've got 30 seconds, as part of the research effort, though, all of those weak genes, although they don't individually tell us as individuals about our personal risk, and certainly not for diagnosis, they put together that jigsaw of causation and also suggest ways, pathways, bits of that chain of event of cause to effect that you might be able to treat. So lots of the genes seem to point to inflammation, the genes in the immune system.
Yeah, yeah.
That, you know, that links up with the story that Mao has been looking at. So genes are very important for research, but I don't want people to go where the idea that most dementia is inherited down in the family.
Right. Yeah, okay, thanks very much. And yeah, that's great. Just also to echo your point about the importance of genetics for picking targets, for drug targets, because there's quite a lot of evidence that if you target your medicine against a gene that is implicated in the disorder, you've got a much better chance of success. So that is also important from that point of view, isn't it? And let's turn now to nurture. And the question here, Zoe, is lifestyle, really. You know, what is it about the way we live, the environment we live in, things that we could possibly change? Does aerobic exercise make any difference? Cold water swimming, doing crossword puzzles? What do we know about environmental effects?
Yeah, so I think as picking up from James' point in terms of the genetics is not the full answer. This is exactly the interplay of lifestyle factors and genetics that can really make a difference. And we know that lifestyle factors can actually by 40% change the pathway of dementia. We've seen recent research that actually shows based on polygenic risk scores, high scores for dementia in terms of high risk based on genetics can be mitigated by lifestyle factors. And I think this is really important research that shows not that the genetics are not important, but actually early on, before the disease, if you want, settles in the brain and causes damage, we can do quite a lot to mitigate through lifestyle factors. And I always say, you know, do as, not as I do, which means sleep more, eat healthier, exercise more. Everything can really help in terms of having that, you know, body-brain balance, and keeping really active. That's really important. We see in dementia, a lot of causes come from comorbidities, for example, from cardiovascular disease. There are factors there that really, lifestyle factors that really can change these causes that then as secondary effect, they might have impact on the dementia trajectory.
I mean, can I ask a sort of slightly personal question about my own dementia modifying behaviors? Because I believe in the inflammation story, and because I've got the kind of teeth that left to their own devices will generate low-grade inflammation. I'm quite obsessive about dental hygiene. Is there any evidence that that is going to make a difference to my long-term dementia risk?
I think there's no conclusive evidence. That there were some stories that I think got everybody excited. And although dental hygiene, I think, is really important, I think it's probably not very closely linked to neuroinflammation. Okay. But I think there are other areas where it can really make a difference. For example, hearing loss
yes,
seems to be
yes. I wonder, can you expand a bit on that? And James, you might want to chip in on this as well.
Yes, as someone who wears hearing aids, it's a subject that's close to my heart. So there's some very important research about 2 years ago from an international Lancet commission looking at what are the lifestyle factors that are linked to the increased risk of dementia, and also that it might be modifiable. And if you were to modify them to deal with those lifestyle factors, how big an impact might you get? And many of the factors on that list were not a surprise. So high blood pressure, obesity, smoking, and diabetes, they were on the list. Their risk was important because they're so treatable, but the effect is quite small. One of the things that came up was actually the biggest single risk was hearing loss, midlife hearing loss. And it's not entirely clear how that leads to the increased risk, but it is something where a lot is known around detecting hearing loss, treating it proactively. Whether it's the biological aspect of hearing or susceptibility of nerves to dying back early, or it's the social impact of isolation. and the impact, is sort of indirect consequences of social isolation. Whatever the mechanism is, it's a modifiable and common factor. And one of the other biggest factors was education. Education in its own right, and education interacting with genes to build a resilient brain that works well throughout your life.
You mean historical education, what you've done, before you develop neurodegenerative disease or aging?
Yes.
Or
so the level of education in childhood, the health and the resilience, the state of your brain as you leave childhood, carries either protection or a risk for the rest of your life.
Okay.
And so that becomes a policy issue as well as a sort of something for individual change. And the other area on the list that is a policy level is very modifiable is air pollution. So some of the lifestyle factors or environmental factors are things we can take personal actions to deal with, what we eat and drink and smoke and so on, our hearing aids. Others are really for us, collective society policy to get to grips with. But if we get it right, about 40% of dementias as a group might be preventable. That's an extraordinary impact with current knowledge.
That is quite a big dent based on what we already know. Let me change tack slightly to an interesting question that's come in that I wanted to ask you, Maura. And it's about, given how anxious people are about the prospect of being diagnosed with dementia, and also the lack of effective treatment, how do you encourage or incentivize people to actually participate in research? Aren't there going to be a lot of people that think, well, do I really want to know? Do I want to know my DNA? Do I want to know if I'm going to get Huntington's disease? Do I want to participate in this research, particularly if there's nothing that can be done yet in terms of treatment? How would you try and persuade people that it's a good idea?
Yeah, that's a very good question. We all share the frustration that there is not a cure yet, and it's very devastating when a diagnosis comes. But research is really improving, and we really need the contribution of everyone. Once that a person goes through the diagnosis process with all counseling and all psychological support, the family support, and social care, I think we really like to incentivize them to take part to our studies, because the only way to get to a cure is to understand what is behind these symptoms, what happens in the brain. And any contribution from people with dementia and their family is so invaluable and unreplaceable for us. So I think to arrive to reach a point where we don't fear anymore to get a diagnosis because there will be a cure, we need any help that we can get from people that already have a diagnosis. And of course, for the genetic, when we spoke about the genetic causes of dementia, when, for example, in clinics, there is a suspicious case that could be related to genetic FTD. Of course, what we suggest is always going through some counseling for testing. And of course, they can decide to get tested or not.
And I wonder what add to that this idea that nothing can be done, I must come in and counter that. So we don't yet have a cure, but I'd agree with that. There's actually an immense amount that can be done, that should be done. And partly if you have memory symptoms, come forward. There are many explanations, only some of which are either dementia or this prodromal run-in period of dementia. There are many other reasons to have memory symptoms and memory problems, which are treatable in their own right. That's the first thing. If you do happen to have those beginning steps of what will later become a dementia, there's a lot you can do around looking after your current health, some medications you might avoid, some you might start, understanding lifestyle measures, understanding how you adapt within your family, within your work, within your time and planning to live well alongside the diagnosis. And there's an awful lot of help and advice to be done to that. It's not just a question of having a diagnosis dumped as a heavy burden to carry. It's around diagnosis as information that's empowering to improve your quality of life and your family's quality of life for the decade afterwards. It's actually really should be seen as a very positive step forward in the right clinical setting. I would admit that does not happen everywhere.
And James,
much to improve.
James, if I could add, you mentioned cancer, and and we have health checks for cancer and for cardiovascular disease, it's really time to be thinking about brain health checks. And this can start very early. All of the neurodegenerative disorders are really a continuum. And if we can start early with the new tools that we are developing and have this early prediction, and 40% of damage we can avoid because we are changing lifestyle interventions, and it's really important to really embrace this, that our brain health is same as our body health.
Indeed, Cambridge is going to be part of a new national network of brain health centres. I know Zoe's very much involved in this, bringing both state-of-the-art, not just technologies around the diagnosis, but actually support, advice and interventions to mitigate any risks that are picked up.
Okay, great. Zoe, you're on the mic, as it were. There's quite a detailed question, interesting question, come in about what kind of data you put into these AI algorithms, and particularly what's the kind of trade-off between going deep, making lots of very detailed, scientifically advanced measurements with imaging and so on, going deep in a small number of people versus just using the kind of clinical data or the social media data that you might have on millions of people. How do you see that trade-off?
Yeah, we need both. We need both approaches. We need the experimental medicine approach that allows the deep phenotyping, getting to the roots, to the causes, being able to record with high precision. And we see this. We have neurotechnology developments that are really amazing. We can now, look very precisely in different areas of the brain. We can measure neuroinflammation markers. If we could also have this data at large scale, then that would be amazing. Really, we will see with the precision, with the power of the algorithms, we would see amazing precision. The challenge there is that we cannot put through this phenotyping a whole population. And therefore, at the whole population level, we have to choose what tools we will be able to really apply to collect this data. And so this choice is really hard, and that's why, because people, of course, that takes quite a lot of time, and at a large scale, we will use some of the approaches, maybe not the most deep phenotyping approaches, but there where we gain is in numbers and also in multimodality. So we collect data that are complementary and come from very different perspectives. Cognitive and clinical data, imaging data, biomarker data, genetic data. And really there what we cover is this heterogeneity in the population, because we need to be able to study these diseases at the level and the scale of populations, people that come with different demographics, different epidemiological backgrounds, different genetic backgrounds, because these are global problems, global challenges that we need to address them at that scale. Usually with deep phenotyping, we cannot really target at that large scale. And so these approaches are very much complementary. The interesting thing is, from the modeling that we do, is we start understanding also disease better, because these large data, as we talked about, they allow us to look at these interactive factors, and they come up with patterns and multi sort of interactive information that gives us new ideas about potentially patterns of disease we have not discovered, and potentially about subtypes, new subtypes that we have not profiled. Then these first ideas about maybe discovering new areas of disease, we can follow with a deep phenotyping, and we can understand better. We can build more precise drug targets. So these approaches really work together, and hopefully this will bring the breakthrough, moving from discovery science all the way to translation.
Well said. And thank you all for a fascinating conversation. I've really enjoyed it. I'd like to thank you for your contributions. I'd like to thank the alumni events team for pulling it all together and making it happen. I particularly like to thank everybody in the audience, particularly those that came up with such interesting questions, and hope you enjoyed it too, and look forward to seeing you in future at another Cambridge Conversation.